In this video, we will be discussing about the bacterial endophytes and its role as plant growth promoting agents and antimicrobial activity against the phytopathogens. So, endophytes are basically the microbes which live inside the plants. They may be a bacteria or fungus. What are the endophytes? Endophytes are the microbes which live inside the plants and colonize in them without having any external sign or negative impact on the host. They live intracellularly as picture shows. This is the colonizing pattern of the microbes inside the tissues. So they have very important role in the host. This picture shows the role of endophytes in the plant development. It is a plant endophyte interaction. So they have a wide array of roles ranging from providing protection to various biotic and abiotic stresses, improving the plant nutrition by production of various phytohormones and secondary metabolites, as well as the removal of contaminants. So the endophytes are very important for the development of host plant. This picture shows the role of endophytes in controlling biotic stresses. So endophytes protect the host plant by induction of systemic acquired resistance or the induced systemic resistance. In addition, they are also able to produce the antimicrobial peptides and proteins to act as a biological control agents and the associated proteins. They protect from the bacterial or fungal pathogens. At an IBSM Raipur, we have isolated the bacterial endophytes from the different plants like chickpea, pigeon pea, lathyrus, rice. The endophytes were isolated from the phyllosporic as well as the rhizosporic region of the plants to see the bacterial diversity across the plant tissues. So the plant tissues ranges from stem, leaf, flower, pod, seed and root nodules. This picture shows the initiation of growth of bacterial endophytes from the different plant tissues. Then the bacterial endophytes were purified by subculturing, and then they were stained using gram staining to see the gram positive and negative reaction. And finally, the bacterial endophytes were identified on the basis of 16S RNA sequencing. So, using this technique, we have isolated more than 200 bacterial endophytes from the different crops. Isolated bacterial endophytes were further characterized for the functional traits. So these traits represents the production of indoleacetic acid, solubilization of phosphate, and the siderophore production activities as well as the DNA assay. Bacterial endophytes were tested for their antagonistic activity against soil-borne fungal pathogens, namely Rhizoctonia, Sclerotium, Fusarium. The dual culture approach was used to screen for antimicrobial assay shows the clear zone of inhibition of fungal pathogens in the presence of bacterial endophytes. On the basis of the inhibition zone and the potential bacterial endophytes have been identified. Bacterial endophytes having antimicrobial activities were further tested for the presence of antimicrobial peptides or lipopeptide genes. It was found that the surfactant, iturin, and bacillomycin D was present in the bacterial endophytes. We have published one paper on the bacterial endophytes isolated from the rice having the antimicrobial activity in the South African Journal of Botany. Pigeon pea bacterial endophytes having antimicrobial activity against Sclerotium rollsii were further validated under in planta condition in pigeon pea and chickpea plant. The result shows the presence of bacterial endophytes enhances the resistance against the Sclerotium rollsii as compared to the control plant. Bacterial endophytes having plant growth promoting activities were in planta validated on the chickpea and pigeon pea plants. It was found that size of root and the leaf were enhanced in the presence of bacterial endophytes as compared to the control plants. The endophytes were also tested for the probable role in the drought tolerance. It was found that chickpea plants having inoculated with the bacterial endophytes have better survivability under drought condition as compared to the control plant. Bacterial endophyte having PGP activities as well as the antagonistic activities against Sclerotium rollsii were in planta validated on chickpea plant. It was found the presence of bacterial endophytes enhances the resistance against Sclerotium rollsii and Also, the enhances survivability of plants under drought stress. This provides a new avenue to develop a multiple biotic and abiotic stress tolerant plant using bacterial endophytes. Thank you.